In drug development, there are four distinct phases of clinical development: Phase 1, Phase 2, Phase 3 and Phase 4, which is usually called post-marketing trials or real-world evidence. Phase 1 clinical trials provide initial safety data to support further testing with larger number of people. As the focus of these studies is primarily safety of investigation rather than efficacy or effectiveness of treatment, the study subjects are frequently a small number of healthy volunteers. A notable exception occurs when a treatment that is designed to be administered in life-threatening disease is known to have severe toxicity. For instance, in Phase 1 cancer clinical trials, the treatment might be first tested in patients whose disease has proven resistant to all other therapies. Pharmacokinetic data might be gathered on both single doses and repeated dosing to assess the rates of absorption and elimination in humans. These kinds of studies might also consider pharmacokinetics in the presence of renal or hepatic impairment, as well as pharmacokinetics in the presence of meals and other drugs. When Phase 1 trial results show unexpected severe toxicities, further consideration of the treatment might be curtailed. Phase 2 clinical trials seek further safety data and preliminary evidence in support of biological effect or efficacy. A slightly larger number of subjects are administered the treatment at a dose or doses that were preliminarily judged safe in the Phase 1 studies. Safety data are collected in a systematic fashion, including specified monitoring of any potential side effects that were identified previously. Phase 2 studies also serve to screen for treatments that show some sign of biological effect, such as a biological marker that is a surrogate for the clinical outcome that is of interest. Products that fail to demonstrate a certain level of biological activity might be abandoned. Phase 3 clinical trials are large confirmatory studies meant to establish an acceptable benefit-safety profile in order to gain regulatory approval for a precisely defined indication. Phase 3 trials are well-controlled trials that provide scientifically credible and statistically strong evidence about the treatment indication hypothesized at the end of Phase 2 investigation. Sample sizes are typically chosen to have a high probability of ruling out the possibility of ineffective therapies and to estimate the treatment effect with high statistical precision. Collection of safety data continues to play a major role, as the larger sample sizes in the Phase 3 study afford a better opportunity to identify relatively rare serious toxicities. Depending on the disease and patient population, anecdotal occurrence of unexpected extremely serious adverse events will often dictate further study of a proposed therapy. Drug sponsors are required to inform providers and patients of toxic effects even when their occurrence is not statistically significant. Phase 4 clinical trials are trials that are meant to evaluate rare but serious effects that occur over time by a new treatment after it has been approved and is on the market. These studies may also aim to evaluate changes in individual behavior that might mitigate the efficacy of the treatment. For example, increased risk-taking behavior when vaccinated for HIV or treated for peanut allergies.